So I'm the black sheep of this presentation because I'm a toxicologist. I have a tiny bit of PK. I used to know how to use Win non-lim, but then they changed the format. So I'm here to talk to you about early tox studies and how you can use what you can do, you know, as an academic or somebody just trying to think about how they're going to translate their research and maybe try to build a company. And of course, I realize that I'm the last person between you and the drinks. And the last person, if you notice, for those of you that live in Houston, are well aware that we have to hurry up and get done with drinks because we have a big game tonight. So I run drug development for Iterion Therapeutics, and I also keep getting drawn into consulting work on the side because there's not a lot of us toxicologists in Houston. So I see quite a few things, and I always, I think it's great. It's fun. I always get to the point where somebody will come in and say, okay, well, it works in mice, and it looks like it's going to be orally bioavailable, so what do I do next? And the answer is, well, you're going to start doing some toxicology studies. And you can start to do studies. I'm going to show you some in vitro screens that you can use as screening tools. So they're inexpensive. If you've got 10 candidates, those are easy to put through. 100 candidates are pretty easy to put through. And then we'll talk about the early in vivo studies that you can do. Those are a little more pricey, so you aren't going to run 100 compounds through those. But one thing that we want to really emphasize is, and you've heard it from other speakers, is that when I used to sit on project teams in large pharma, my goal wasn't to get a drug to jump over a fence and get it ready for the next stage of development. My goal was to kill a drug before it went into development, kill it early on so that we could move on to something else. And that's easier to do in large pharma because they have a pipeline. It's harder to do if you're a small company and you come in with me and you're like, well, this is all I have. So then you're a little bit more trying to tell me how can I get it over this fence of it did this bad and it did this. So the goal isn't really to show that a drug is safe, it's to show that a drug is toxic and to try to kill a drug. So I also see examples of where somebody will come in and they'll say, okay, it works, and so I've outlined this study that I think will get me into IND. And it's a rat study, and it's a single dose, and because I'm going to use standard of care on top of it, I have a dose group that is the clinical dose by itself for the drug, and a clinical dose plus the standard of care, and then the relevant controls. And if I show that it's safe, that nothing bad happens when I use that dose in a combination, I'm good to go, right? And that's not it, because the goal of a toxicologist isn't to show that the drug is safe. The goal of the toxicologist is to define what the toxicities are, because I'm going to be the one sitting in the room when you're dosing that first patient going, man, when you do the clin path tomorrow and the BUNs or creatinines are up, we know that that's a toxicity that I've seen in the animals, and you probably need to tell them they're not getting the second dose. So I really want to know what the target organs are for toxicity. I want to dose high enough that I see that. I'd like an idea of the dose response curve, so I'm going to use multiple dose levels, because if I have a compound that goes from 0 to 60 and at one dose it's safe, nothing really happens, and I double the dose and the animals are all feed up in the cage, that's a scary drug for me. And I'm probably going to say, man, I can't deal with that kind of dose response. So I want to know an idea of how, you know, what the therapeutic index is and how fast you go to really killing all the animals from seeing a few signs. And then probably in early studies, I'm not going to worry about reversibility. But when I get into GLP tox, I also want to know whether or not what I'm seeing is reversible. Because some toxicities, like doxorubicin in the heart, aren't. And so the clinician will keep track of how much you've got. And when you hit a certain dose, that's not an option for you anymore. But a lot of them are. So, you know, chloroform in the kidney is pretty reversible. A lot of those are reversible. And so if I know that, and so if it's to tell the physician, look, if you take them off, the patient doesn't have to panic because the BUNs will be down to normal when you bring them back in 3 days. And then everything is good. So I say that we want to kill drugs early because we've also seen this up here, and this is kind of an old slide, so it's probably underestimated cost. But it probably will take you $1 million to get a drug into your IND. And so that's a lot of money. It'll probably take you $3 million to do these later studies, and we are going to talk about these later studies, because you really aren't going to have these done. And if you're a small, if one of you guys is going to fill out a company, the chances of you actually having to run this seg2 teratogenicity study are probably slim, because you're going to license it out when you show it phase 2 proof of concept. So you're really only going to have to worry about this $1 million. And you're going to spend most of your money on these 4-week GLP tox studies. So we're not going to talk about tox studies, the GLP tox studies today. ICH, so International Conference of Harmonization, and that was probably one of yours in your appendix, right, where you listed all the sites, Johnny, saying, oh, go to this. So ICH has really nice guidances on how to, the length of your study. It's generally 4 weeks is the default to get your IND tox studies, and then I'll talk a little bit about what you need to do for those. But between that set of studies and doing your gene tox battery, and ICH defines that in one of their guidance documents, just like a cookbook, ICH also defines what they want you to do in terms of your safety pharmacology battery. If you do those set of studies, those probably are on the matter of $700,000 to $900,000. So most of your cost is going to be, of that $1 million, is going to be on these GLP studies, gene tox by GLP, and so you want to kill it early with the studies that we're going to talk about, because you're going to go out and try to raise investor dollars, and $1 million is a lot. So I got 2 slides on some of the in vitro screenings you can do that are commonly used. The first 2 are the HERG assay and an Ames assay, and so a lot of times when you go out to a little CRO and you're doing your in vitro ADME package, and you're doing your plasma stability and microsomal stability and your in vitro hepatocytes, this is just an add-on, and they can do it at the same time. It's probably about $1,000 each to run. You get a little bit of a discount the more compounds you put into it. But FDA is pretty concerned about, a positive HERG. ICHS7 talks about that. They're kind of concerned about whether or not a compound is genotoxic. They talk about that in ICHS2. So I always get somebody, we run this assay, and it's a positive HERG. And they're freaking out and say, what are we going to do? And so it kind of depends, right? It helps if you, before you really start down the road of doing something, if you write up a target product profile, and nobody's really talked about that, but when you start and you sit down, you should say, what are the attributes that my drug has to have for it to be successful in the space that I want to go in? And that's my target product profile. And you might say, look, in large pharma, if I had a drug for pediatric asthma, one of the things I might write is no cardiovascular liability. So if I see a positive HERG, it's done. I'm not going to try to get it over a fence to say, okay, well, I can deal with this. But I have companies that come in and like say, I have a positive HERG, and I have a couple of indications I can go into. And it's like, well, it's not a killer if it's end-stage cancer to have a positive HERG. And obviously, it's not a killer to be mutagenic if you're a cancer compound, because the general cytotoxics are all mutagenic. It depends on the risk-benefit. It depends on where you think you're going with your indication. So if I had a mutagenic compound and I thought I was going into obesity, my target product profile is going to say, yeah, I can't carry this compound over that fence for that. And that's a good way to kill it. So it's all a risk-benefit as to whether or not you see a positive in something. But it's a good way to screen compounds, and it's relatively cheap. So the other thing you'll see a lot of people talk about is this safety screen 44. And you can also do a safety screen 76, I think it is. But it's offered by Eurofins. They're one of the only ones that do this. But they have a panel of 44 targets, pharmacological targets, and you're looking to see whether or not you have any off-target effects. And everybody always panics on this too, and they say, oh my God, I got a result like this. And I had my compound had inhibition of 2 targets at greater than 50%. And this assay is run at 1 concentration. It's run at 10 micromolar, and it looks at inhibition at different targets. And then the company says anything over the 50, you should think about what it means. So you think about what it means. So maybe I have 4 targets, and they're all CNS targets. And I know from my PK studies that it doesn't cross the blood-brain barrier. So I say, what do I care? has those targets. I might get one where I've had like, maybe I had a negative hERG, but I have like 4 ion channels in this panel that pop up as a positive. Now these are binding assays, so they aren't functional. So I've had this with a couple companies where I get like 3 of the ion channels are targets on here, and then we go back and I run a in vitro patch clamp assay for the different channels to do a functional assay, and then it doesn't do anything in a functional assay. So then you're okay, but it's just this running this off-target safety screen gives you ideas of, hey, maybe I should look at something along here. So if I saw a positive of these 2, the next thing I do is probably run an IC50 because I want to know the dose response curve for that, and then I think about what it might mean for my molecule. And that one costs about 6 grand, so you probably aren't going to put a ton of money or a ton of compounds into that. That money goes up fast. So dose setting. So now I've done my in vitro stuff, and that doesn't chew up very much compound. Now I'm getting ready to think about, okay, I want to do rats and I want to do dogs, but all I have is, maybe I have the first rat study, but you know, you aren't trying to kill a rat in your PK study, so maybe you probably dosed it a kind of couple lower doses, so you don't really know the toxic doses. In mice, you have an idea of what the pharmacological efficacious dose is, so you're thinking, what do I need to do to pick my doses? Because compound is probably scarce at this point, and especially dogs chew through compound really fast. So, you know, you could say, hey, I can make you 100 grams of drug and have it ready for you, but that's probably all I can make if there's 3 months. But 100 grams, that's a buttload of compound. I mean, you've been using milligrams in vitro, so, but you start to do the calculations. You can scale from mouse to dog, and you can say, well, you know, we probably have to start at 1 milligram per kilogram in dogs, and we'd really like to get to, you know, FDA really likes a 25, around a 25-fold safety factor, so I'd really like to get to 25 mg per kg in my dog. And 25 mg per kg, 10 kilo dog, 250 milligrams, and I'd like to dose for 7 days. The drug goes really fast. And so you start to think about, what does my study look like? And how do I set my doses? So sometimes I'll I'll always set the bottom dose to say, what's the dose that works in mouse? And I can scale that. There are guidance documents on FDA that tell you how to scale. I scale the dose on a body surface area. What is the dose that I think would be pharmacologically active in a dog? And I'll probably add maybe a factor of 10 onto that and say, okay, that's what I want my low dose to be. And then my mid and my high dose will be any kind of multiple of that. If I had unlimited drug, I'd like to get, we like log multiples, so we like 3 times the clinical dose, and then 10 times the clinical dose, and then 30 times the clinical dose, and if I could get to 100, that'd be great. But I really, 30 FDA is pretty happy with. But there are a lot of considerations. Do I have enough drug? Is it soluble in the dose of volume that I need? Sometimes I get people that are like, I got this really great sub-Q product, and the solubility is 10 mg per mil. And I need a dose of 1 mg per kg. And so what do you think? And it's like, that's like 10 mils in somebody's arm. It's going to be a non-starter. And I can't do that in a dog study either. So sometimes you're limited by volume considerations. Sometimes you're doing, you probably aren't doing inhalation for any drugs you're doing here, but inhalation is nasty. It chews through drug at a phenomenal rate. So I have a company now that I'm working with that we're really trying to fight, because the FDA may come back and say we'd like 14 weeks of daily exposures, and they're like, we can make 4 weeks. And that's all the drug we have. And luckily it's antigenic, so at 28 days the antibodies are pretty high, so the argument is that the antibody response will make continued dosing past 4 weeks unnecessary. But you can do a lot of things, but it's an art form. There aren't really any guidances that say you have to do exactly this to do your dose setting. Sometimes you get lucky, sometimes you don't. It's a lot better to be unlucky in the rats. So tox studies are, the non-rodent species always rats. It's a lot better to be unlucky in here because people don't really mind so much having dead rats in your high dose group. At least you define the MTD. But we use rats instead of mice. One, FDA has a lot larger historical database on rats. So say I see a histological finding that's like, haven't never seen that before. And it's it's always in the high dose group by Murphy's Law. But maybe it's in the mid dose group and you'd like to say, does FDA have a knowledge of maybe they've seen this? Or the contract lab have a knowledge that they've seen this at a really, really low incidence, but they have seen it in control animals. You know, you get that kind of data because they have that data for rats. And you don't have that data so much for mice. The other practical consideration is we talked about you'd like to do some PK sampling, and most people don't have the method up and running to do only 20 microliters. They have to do 100 microliters, which means killing an animal at every time spot. So instead of using 6 per sex for your satellite PK groups, you're up to 30 per sex for your satellite PK groups. And then you're doing clinical pathology at the end of the study. And the blood volume that you need for clinical chemistry takes up an entire mouse. And then the blood volume you need for hematology takes up another mouse. So you'd have to double the amount of mice that you have in your main study to get enough ClinPath data. So nobody likes mice. Everybody uses rats. So rats in this dose range finding study, I always start with maybe 3 to 5 per sex. Want to do at least three dose levels in a vehicle control. And then my dose duration might be determined by how much drug you tell me that you have. I'd like to get at least 5 days. I've started to see drugs that I see my toxicities around day 10 and 11, so it's made me gun shy, and now I tell people I'd really like you to have 14 days just to be safe, because the last thing you want to do is run a 5-day rat study, start your GLP tox study, and 10 days into it, the high dose group is all dying, and you have to decide, okay, so can I lower my dose? Should I just terminate them all now? You know, what do I do? So it's better that you do that up front and do it a long enough duration to get answers. I'll do histopath. It's pretty expensive, so I'll do the high dose. I'll tell the contract lab or whoever's doing it, hey, take all the tissues out of the controls in the high dose, run those, process them all, look at microscopically, save the other tissues from the low and mid dose into the formalin buckets, and I'll pick out my target organs from the high dose and then look at those in the lower and the mid. So if I see something in the spleen in the high dose group, I kind of like to know what other doses it's seen at, so I'll pull the spleens from the mid dose and the low, and then I'll look at those. And then I'd also like to get PK, because especially, you know, you probably as a PK person are telling me, well, I've already got the PK, but you probably have it really for single dose. And I'd like to know, say, well, I'm going to dose 14 days. So tell me what the PK looks like on single dose and on 14 days, so I have an idea if there's accumulation, what happens. In this study, if you run it about 5 days and you find a good lab, you could probably do around 10,000. If you went out to 14 days, probably take you 20,000. Then you have to add the cost of the bioanalytical running samples at $100 a sample, so it probably adds another 7,000 to 8,000. But still, 30,000 to get you into the study that costs you about 300,000 for the GLP tox. So, dog study, it's always beagle dogs. It's not strays that they catch off the street. When I, tell people I'm a toxicologist, they all lock their dogs up and say, oh my God, he's going to come and get my dog in the middle of the night because it's a lot more cost efficient. And it's like, no, we use beagles. We use beagles that have never seen the light of day except, from the transport through the cage bars. They're purpose-bred for that. So it's always beagles because they have a great historical background, right? So they the genetic background of them. The last thing you want to do, really, pull somebody's dog off the street that you don't know what they've eaten, you don't know if they've heartworms or something, and then you see a toxicity in one of the organs, and you're like, oh, I don't know what that's due to. So no, we don't pull dogs off the street. We use regular old dogs. But we don't like to kill dogs, so I don't mind doing the rats at all. But the dogs are a little bit more difficult because they're beagles, and I used to raise beagles. In fact, I'm getting ready to start a 10-day beagle study because we're planning on doing juveniles in the clinic, and I know that I start them at 10 days, and the techs after 8 weeks they're gonna find it really hard to terminate those dogs. So we don't like doing beagle dogs. So we do the beagle dog study, especially this first one. We do it differently than the rats. So the rats we can start dosing all at one time.
topic came up in terms of a GLP study and the age of the dog, full juvenile.
Yeah.
Any experience on what's in?
Yeah. So remind me when I do this, because I have a good study from the monkeys and the AMD. So we'll come back to the age of the dogs. But so for the rats, we'll set our low, mid, and high, and we'll start dosing all on the same day. Because if I lose the high dose group, I lose the high dose group. For the dogs, I'll start in 1 or 2 per sex at a low dose that I think is going to be tolerated. It's one of the doses that I back calculated from the mouse efficacy dose. And I'll dose those dogs, and I might dose those for 5 days at that dose level, make sure everything's okay, and then I can either take those same 2 dogs and I can wash them out for 3-4 days, whatever the half-life is, get a sufficient washout, and then escalate them up to the next dose. Or if I want to go a little bit faster, I can have multiple doses of dogs, 1 to 2 per sex, and say after the first dose, I get 5 days into it, and they don't have any signs, and the clin path is clean. I'll start the next dose, and I'll dose at that level for 5 days. I'll do clin path if that's clean. Then I'll come back either to my first dose or I'll add a 3rd cohort. But I'm not just throwing my high dose at the dogs and saying, well, handle it, or you don't. I'm trying to kind of escalate. So when the dogs are looking bad, they aren't eating, or they're throwing clin path changes, I can say, okay, that's the MTD dose. And then I'm done with this study. Although usually I'll take that high dose group and I'll probably terminate them and do histopath just so I know what the organs are. But I'm really trying to save dogs. So if I only do 1 to 2 and I escalate the same dose of dog, same dogs through different dose escalations, I can probably get that study done for 10 grand. And another 5 for the PK. So it's somewhat inexpensive, but if I want to go to 14 days and I want to do 6 dogs per sex because I want to use naive dogs at each dose level, because the only bad thing about if I say I'm going to escalate the same group of dogs is I dose them 5 days, I wash them out for 3 days, dose them another 5 days. I'm not entirely certain that the effects that I might see at the end of that second 5-day interval weren't cumulative from the first dose. So there's a chance that I overdose dogs when I do the GLP tox study. But it's animal numbers to compare. So I did 20 minutes. So use your in vitro assays to get rid of your dogs, your warts, I should say. Get rid of the compounds with the warts first, because you don't want to try to carry those over. They can be run along with your other in vitro ADME. Identify problem areas from your in vitro screening. So say I saw some of those CNS effects, and it does cross the blood-brain barrier. I've seen one of those compounds. So you would just say, when you do the first rat study, you might say, hey, I want to do an FOB assay, a functional observational battery, because I think there might be some CNS effects. And I've done that where you put the rat on the table and it's like, wow, he just moves, like he keeps his butt in one area and moves around in a circle. I know that's probably CNS, so my syrup study was right. It's like, yeah, okay, you guys have to send me a different compound. So identify problem areas. And then do your early studies, rats and dogs. Get your study, get your dose levels to help you set your GLP tox doses.
I have a quick question, but it's a daring question. Forget about the FDA guidance. And
I wish we could.
This is for my edification, okay? Yeah. It's have you seen people using the Mabel way to calculate the first human dose? Okay.
Usually, if it's a cancer drug, that starting dose is in the ICHS-9, and it's a little bit different. It's based around a 10-fold factor around the STD, the dose that kills 10% of the rats, with a safety factor of 10, or the highest non-toxic dose in dogs with the safety factor of 6. In the other indications, man, it's just safety factor of 10, and whatever species is the most sensitive is the one that it's set on.
I'll go first. Thank you for the very comprehensive review. My question is regarding plasma protein binding property. I mean, I do in vitro studies. It's very important for us to consider the toxicity or potency.
Right.
But it doesn't seem to be even a consideration when you design the dose. So
it's absolutely no consideration. So our first speaker, when he said protein binding, don't give a rip about protein binding. Because look, a lot of times in your in vitro assay, so I worried about that once upon a time, and I was like, what the? And then I realized that she's putting in a BSA into the cultures for the pharmacological activity. And it's like, well, you're putting protein in the cultures, and it's working at 10 micromolar, or whatever micromolar it is. It'll work in the animals the same way. And we're not talking about covalent binding, so there's this equilibrium, right, between it coming on and off that protein. So I never try to adjust dose on the basis of protein binding.
So what's the usage of that piece of information?
You guys, I'll say you want it. Because it's, you know, you do that, you run the PBPK, the little in vitro ADME, and everybody wants to know what is protein binding. Even the clinic, you know, as we get into clinical studies, I've never had anybody say, hey, this is going to be co-administered, these are the con meds, and is there any concern? Because the concern would be like I'm going to knock the one that's protein bound off the other maybe, but even that I don't see, you know, nobody really ever asked me, hey, the standard of care is 99% protein bound and your protein binding is 90%, do you think you're going to have to adjust the dose? And they only really do that if it's like a CYP3 or there's some kind of inhibition or induction of a microsomal and they both share the common metabolic pathway.
Is there a big difference or small difference between your study design for different diseases such as cancer or infectious disease, like when you design your tox study? Is that?
No, they're pretty agnostic.
So it's standard. Right,
it's pretty standard, low, mid, and high. No, so I said that 4-week study is like a standard IND. If I have my clinical team say, look, we're going to start in phase 1B dose expansion, we're going to dose for 6 weeks before we get an idea of a clinical marker, I might say, yeah, why don't we make it 6 weeks instead of 4 weeks, just because I know the clinical team is telling me that patients are going to be dosed for 6 weeks.
So those parameters are pretty similar. How about the species you choose?
Yeah, so the species is always going to be rats and dogs unless it's like a biologic. Okay. Or unless the metabolic pathway is such that, look, the dog throws this special metabolite, so I'm not using the dogs. Or the monkeys look better, the metabolic profile looks better in monkeys. But if I could cover the main metabolites in rats and in dogs, and monkeys had both of them at the same time, I would still do rats and dogs because I could say, look, I've qualified that metabolite in rats and I've qualified this other metabolite in dogs. Because the reality is that we don't really want to use monkeys in research because they're really expensive, and FDA doesn't want us to use monkeys in research because, you know, beagle puppies pop out 6 to 10 at a time, and the gestation period is low, so I can generate beagle puppies really quick. But to generate cyno monkeys, it's 6 months, and there's a 10 to 20% stillbirth, and it takes me a long time to generate monkeys. So you might say, oh, well, let's just do monkeys, but FDA doesn't want you to because, you know, otherwise we have a shortage of monkeys, and the studies that really need monkeys won't have them then.